My name is Karen, and I'll be talking about the control of the cell cycle. Before going over how the cell cycle works, we should know more about the cell division of mitosis. Mitosis is to make another copy of itself as a result. The first phase of mitosis starts out with interphase, and this is where it prepares for cell division to occur, and this includes the G1 phase, the S phase, and the G2 phase. Next, prophase would have the cell divide and have the chromosomes start duplicating. Prometaphase would bring the chromosomes toward the middle, and metaphase has the chromosomes aligned in the center of the cell. Anaphase would start splitting the chromosomes, and telophase would separate half the chromosomes to one side and half to the other. And finally, cytokinesis would then result to two identical cells with the same amount of chromosomes. Now that we have the basic knowledge, we can move on to the cell cycle. Cell cycle is the process to replication, producing two daughter cells as a result. And the beginning of this phase starts with the interphase, as we mentioned during the mitosis, and again it includes the G1 phase, the S phase, and the G2 phase. The G1 phase is the beginning phase where the cell grows, and S phase would have the cell continue to grow, copying chromosomes during the process. G2 phase would then have the cell enter cell division, and two daughter cells would be produced after going through the mitotic phase and the cytokinesis phase. During the cell cycle, the G1 phase is the most critical checkpoint where the cell would be able to go through the process of dividing if the cell receives a go sign from these checkpoints. The G2 and the M checkpoint would be able to receive the signal and continue the process as long as they get the go sign. Internal signals would have promoting factors, while external signals would have growth factors. However, if the cell receives a stop sign, it would exit the cycle and switch to the G0 phase, which is a non-dividing state. Next, we will talk about the cyclins and the cyclin-dependent kinases. Cyclins are where there's a huge amount of proteins that will help advance to the next stage. If you look at the gap between each phase in the graph, the G1 phase, the S phase, the G2 phase, and the mitosis phase, there's a set of proteins that would build up. An example would be the cyclin A, where it would build up and accumulate more cyclins over each phase. The cyclin-dependent kinase, also known as CDK, is a chemical that can speed up actions within a cell. It is dependent on the accumulation of cyclins, which would help by fitting into the CDK and activate it to do certain things, such as the mitosis promoting factor, also known as the MPF. The MPF is an internal signal that builds up enough kinases that will work on the cell. It would cause to do things such as breaking the nucleus and working on microtubules, pushing the cell into the mitotic phase to produce two daughter cells. As for an external signal, an example is a platelet-derived growth factor, also known as PDGF, and this is made by blood cell fragments called platelets. This is a growth factor that allows the cells to pass the G1 checkpoint and divide. PDGF would stimulate fibroblast division not only in artificial conditions of cell culture, but also in an animal's body. When there's an injury occurring, platelets will release PDGF, resulting to proliferation of fibroblasts to help heal the wound. Finally, we'll be talking about the cancer cells. If there's a problem with the process of cell cycle, or it doesn't occur, this is where cancer takes place, where the cell division continues to divide and divide. The problem would begin when the normal cell, undergoes transformation, leading to the tumor cells, where, like I said, it would turn the normal cell into a cancer cell. The human's immune system would be able to recognize and destroy the cell, but if the cell happens to evade it, it would result to proliferation and leading to tumor growth. And there are two kinds of tumors. There is the benign tumor, which is not serious and can be removed from surgery, and the malignant tumor, which can be harmful enough to impair functions of one or more organs. If someone gets a malignant tumor, it can be invasive enough, leading up to the tumor metastasis, and this is the spread of cancer from the original state. And this is all I wanted to say about the control of the cell cycle, and I hope that was helpful.